Welcome. Today, we're taking an inside look at the most complex organ in the human body, the brain. Let's dive into its gross anatomy.
To understand the brain's organization, we divide it into four major regions. First, the large cerebral hemispheres. Second, the centrally located diencephalon. Third, the brainstem, which consists of the midbrain, pons, and medulla oblongata. And finally, the cerebellum, located at the base, responsible for coordinating movement.
Before we trace the flow, let's define the ventricles. These are a connected system of four fluid-filled cavities located deep within the brain parenchyma. Rather than being solid tissue, these spaces serve as the production site and reservoir for cerebrospinal fluid, providing an internal cushion that protects the brain from within. The ventricles are a connected system of four hollow spaces within the brain, filled with cerebrospinal fluid, or CSF. This fluid is constantly produced by the choroid plexus found within these cavities. The journey begins in the two lateral ventricles, flowing through the interventricular foramen into the third ventricle. From there, it travels through the cerebral aqueduct into the fourth ventricle. The fluid then exits through the median and lateral apertures to enter the subarachnoid space, circulating around the brain and spinal cord. Finally, it is reabsorbed through the arachnoid granulations into the superior sagittal sinus, eventually draining into the internal jugular veins to return to the blood.
Next, we examine the cerebral hemispheres. The surface is covered in folds called gyri, separated by shallow grooves known as sulci. Major landmarks include the central sulcus, the lateral sulcus, and the Parieto-occipital sulcus. Deep grooves called fissures further divide the brain. The longitudinal fissure separates the two hemispheres, while the transverse cerebral fissure separates them from the cerebellum. These landmarks define the 5 lobes: the frontal, parietal, occipital, and temporal lobes, along with the insula, which is tucked deep inside. Finally, on the inferior surface, we find the olfactory bulbs and tracts, responsible for our sense of smell.
Now let's identify the external functional areas of the brain. Starting in the frontal lobe, we have the prefrontal cortex, the primary motor cortex, located on the precentral gyrus, and the somatic motor association area, or premotor cortex. Moving to the parietal lobe, we find the primary sensory cortex on the postcentral gyrus, followed by the somatic sensory association area. For our senses, locate the auditory cortex and its auditory association area in the temporal lobe, and the visual cortex with the visual association area in the occipital lobe. Finally, we identify the specialized centers: Wernicke's general interpretation area, Broca's speech center, and the frontal eye field.
Moving inside the brain, we find the internal anatomical structures. First, locate the corpus callosum, the massive white matter bridge connecting the hemispheres. Just below it is the fornix and the thin septum pellucidum, which separates the two lateral ventricles. Within the ventricles, look for the dark, knotted choroid plexus, responsible for CSF production. Deep within the white matter, we identify the basal nuclei, or ganglia. This includes the arching caudate nucleus and the lentiform nucleus, which is composed of the lateral putamen and the more medial globus pallidus.
Now we'll examine the diencephalon, starting with the external structures visible on the inferior surface. First, locate the visual pathway, the optic nerves, leading to the X-shaped optic chiasm, and continuing as the optic tracts. Moving posteriorly, we find the paired mammillary bodies, the pineal gland, tucked toward the back, and the infundibulum, which serves as the stalk connecting to the pituitary gland. Shifting to the internal structures, seen in a sagittal view, we identify the large, egg-shaped thalamus, which contains the small interthalamic adhesion connecting the two sides. Surrounding this area is the slit-like third ventricle, where the choroid plexus produces cerebrospinal fluid. Finally, we locate the hypothalamus, forming the lower walls, and the epithalamus, forming the roof.
Now, let's transition to the brainstem, starting with the superior-most region, the midbrain. On the anterior surface, identify the cerebral peduncles, and internally, the cerebral aqueduct. Turning to the dorsal surface, we find the corpora quadrigemina, consisting of the superior colliculi and the inferior colliculi. Emerging from this region are the oculomotor nerve and the trochlear nerve. Next, we move down to the pons. Here we identify the trigeminal nerve, the abducens nerve, and the facial nerve. Finally, we reach the medulla oblongata, the most inferior part of the brainstem. Along the midline, locate the decussation of the pyramids. From here, we find a sequence of cranial nerves: the vestibulocochlear, the glossopharyngeal, the vagus nerve, the accessory nerve, and the hypoglossal nerve. Finally, we examine the cerebellum, or little brain, located posterior to the brainstem. Looking at the external structures, we see the right and left cerebellar hemispheres, joined in the middle by the ridge-like vermis. The surface is composed of thin, leaf-like folds called folia, which are divided into the anterior, posterior, and the deep flocculonodular lobes, separated by the primary fissure. Shifting to the internal structures, seen in a sagittal section, we find the beautiful branch-like white matter known as the arbor vitae, or tree of life. This is surrounded by the outer cerebellar cortex, with deep cerebellar nuclei embedded within. Lastly, we note its relationship to the fourth ventricle and the choroid plexus, located just anterior to the cerebellar mass.
To protect the delicate neural tissue, the brain is encased in 3 layers of membranes called meninges. The outermost, toughest layer is the dura mater. It consists of 2 layers, the outer endosteal layer, which attaches to the skull, and the inner meningeal layer. Between the dura and the next membrane is the subdural space, which is considered a potential space. The middle layer is the arachnoid mater. Beneath it lies the subarachnoid space, which contains cerebrospinal fluid. You can also see the arachnoid granulations, which look like small tufts that return fluid to the blood. The innermost, microscopic layer clinging directly to the brain surface is the pia mater. The dura mater also forms protective folds that dip into the brain's fissures. These are the dural folds. Locate the falx cerebri in the longitudinal fissure, the falx cerebelli between the cerebellar hemispheres, and the tentorium cerebelli, which acts like a tent over the cerebellum. Finally, identify the venous channels where blood collects, the superior sagittal sinus along the top, and the inferior sagittal sinus, running just below it.
Now, let's identify the 12 pairs of cranial nerves by their Roman numerals, locations, and functions. Associated with the cerebrum. Cranial nerve 1, the olfactory nerve. Located on the inferior cerebrum, it is sensory for smell. Cranial nerve 2, the optic nerve. Located at the diencephalon of the cerebrum, it is sensory for vision. Associated with the midbrain. Cranial nerve 3, the oculomotor nerve. Located on the ventral midbrain, it handles eye movement and pupil constriction. Cranial nerve 4, the trochlear nerve. Located wrapping around the lateral midbrain, it controls superior oblique eye movement. Associated with the pons. Cranial nerve 5, the trigeminal nerve. Located on the lateral pons, it provides facial sensation and motor control for chewing. Cranial nerve 6, the abducens nerve. Located at the junction of the pons and medulla, it abducts the eye. Cranial nerve 7, the facial nerve. Located at the pontine medullary junction, it controls facial expression and taste. Cranial nerve 8, the vestibulocochlear nerve. Located lateral to the facial nerve at the junction of the pons and medulla, it is sensory for hearing and balance. Associated with the medulla oblongata. Cranial nerve 9, the glossopharyngeal nerve. Located on the lateral medulla, it handles taste and swallowing. Cranial nerve 10, the vagus nerve. Located on the lateral medulla, it provides parasympathetic control to visceral organs. Cranial nerve 11, the accessory nerve. Located on the lateral medulla and spinal cord, it controls neck and shoulder muscles. Cranial nerve 12, the hypoglossal nerve. Located medially on the medulla, it is motor for tongue movement.
To help you master the cranial nerves for your exam, let's use two classic mnemonics. First, to remember the names in order, use the phrase, "On occasion, our trusty truck act funny. Very good vehicle anyhow." Cranial nerve 1, olfactory, for on. Cranial nerve 2, optic, for occasion. Cranial nerve 3, oculomotor, for hour. Cranial nerve 4, trochlear, for trusty. Cranial nerve 5, trigeminal, for truck. Cranial nerve 6, abducens, for act. Cranial nerve 7, facial, for funny. Cranial nerve 8, vestibulocochlear, for very. Cranial nerve 9, glossopharyngeal, for good. Cranial nerve 10, vagus, for vehicle. Cranial nerve 11, accessory, for any. Cranial nerve 12, hypoglossal, for how. Now, to remember the types, sensory, motor, or both, use the phrase, "Some says money matters, but my brother says big brains matters more." Cranial nerve 1 and 2 are sensory. Cranial nerve 3 and 4 are motor. Cranial nerve 5 is both. Cranial nerve 6 is motor. Cranial nerve 7 is both. Cranial nerve 8 is sensory. Cranial nerve 9 and 10 are both. Cranial nerve 11 and 12 are motor.
Thank you for watching. We hope this supports your studies. Don't forget to like and subscribe to stay updated. See you next time.